What if I told you that the most powerful cancer-killing drug being used in hospitals right now, the drug that has saved millions of lives across the world, came from a tree that loggers threw away? A tree they called useless, a tree so small and twisted that nobody wanted it, a tree that almost got wiped off the planet before we even understood what it was hiding. And here is the part that will really get you. This is not a rumor. This is not a conspiracy theory. This is documented, verified, peer-reviewed science, published by the National Cancer Institute. Approved by the FDA, used in hospitals today. This is the real story of how two forgotten trees, one from the forests of America, one from ancient China, changed cancer treatment forever. And nobody taught you this in school. Stay with me, because this story is going to change how you look at every single tree you walk past for the rest of your life. August 21st, 1962. Washington State, USA. A botanist named Arthur Barclay is making his way through the Gifford-Pinchot National Forest, a dense stretch of old-growth trees near Mount St. Helens. He works for the U.S. Department of Agriculture under contract with the National Cancer Institute. His job is simple and exhausting: collect plant samples, label them, send them to a lab, repeat. That day he stopped in front of a small, scrubby, unremarkable tree, the Pacific Yew, scientific name Taxus brevifolia. Loggers across the Pacific Northwest hated this tree. It grew too slowly. Its wood was too twisted to use for lumber. It was small, tangled, and hard to work with. When big logging companies cleared forests, they left the Pacific Yew behind or just burned it out of the way. It was considered a weed tree, a trash tree, completely worthless. Barclay was not a tree expert. He was primarily a shrub and seed man. But that day, following the protocol of his program, he stripped some bark off the Pacific Yew, threw it in a collection bag along with some needles and twigs, labeled it, and tossed it in the back of his truck. It was one of roughly 200 random plant collections he had made that summer alone. He drove away and never thought about it again. That bag sat in storage for 2 years, 2 full years. Meanwhile, at the Research Triangle Institute in North Carolina, a medicinal chemist named Dr. Monroe Wall was doing something almost impossibly tedious. He was going through thousands of plant extracts, one by one, testing each one for cytotoxicity. That means testing whether something is poisonous to cells, specifically cancerous cells. Most samples were nothing, dead ends, weeks of work for 0 results. Then, sometime in late 1963 and into 1964, Dr. Wall opened a batch of extracts from the Pacific Northwest. He ran the test. The yew tree bark extract was powerfully cytotoxic. It was killing cancer cells in a way that was unusual, different from anything he had seen before. Dr. Wall flagged it immediately. He requested more bark samples. He and his colleague, organic chemist Dr. Mansukh Wani, began working to isolate exactly which compound in the bark was responsible. It took years of painstaking work. In 1967, they finally published their findings in the Journal of the American Chemical Society. The compound had a name now. They called it Taxol, a play on the tree's genus, Taxus. That discarded trash tree had been holding one of the most powerful cancer-fighting molecules ever found in nature for hundreds of years, while loggers burned it, while nobody paid any attention. Still with me? Good. Because what happened next almost killed this discovery completely before it ever reached a single patient. By 1977, the National Cancer Institute had confirmed Taxol's anti-tumor activity in animal models. It selected the drug for clinical development. Human trials were coming, but there was a problem, a devastating, almost unsolvable problem. To produce enough Taxol for one single patient's full course of treatment, you needed to harvest the bark from 3 Pacific Yew trees. And here is the brutal part: harvesting the bark killed the tree. A Pacific Yew tree takes up to 200 years to reach full maturity. You need 3 per patient. There are hundreds of thousands of cancer patients who needed this drug. Do that math. It does not work. In 1977, the NCI placed an order for 7,000 pounds of Pacific Yew bark. That meant cutting down 1,500 trees that had been growing in old-growth forests for centuries, scattered across millions of acres of protected land. Environmentalists took notice immediately. The Pacific Northwest was already on fire over the northern spotted owl, a protected bird living in exactly these forests. Loggers and environmentalists were already in a bitter, decades-long fight over whether to protect these ancient ecosystems or open them to industry. Now the federal government wanted to tear them apart for medicine. It became a national crisis. Scientists needed the bark, cancer patients needed the drug. Environmentalists needed the forests to survive. Every group had a completely valid argument, and the numbers only got worse. Clinical trials were producing astonishing results. A 30% response rate in patients with advanced ovarian cancer who had already exhausted every other treatment option. 30% for people who had been told there was nothing left to try. For people who had gone home to say goodbye, word spread fast, demand for Taxol exploded. Scientists ran the numbers. To treat every ovarian cancer patient in the United States alone, they would need to harvest 360,000 Pacific Yew trees. 360,000 trees, each one up to 200 years old. There were not enough trees in the world. The drug that could save millions of people was going to destroy an ancient ecosystem in the process. And even if you destroyed the ecosystem, there still would not be enough. It looked like a dead end. This is where the story turns, because scientists refused to give up. And one discovery in France changed everything. Chemists around the world understood the stakes. Taxol worked. It worked better than almost anything else available for ovarian and breast cancer. Giving up on it was not an option. Finding a sustainable way to produce it without harvesting wild trees was now a scientific emergency. The breakthrough began in France. A chemist named Pierre Potier discovered that a compound called 10-decetylbicatine, found in the needles of the common English yew tree, could be chemically converted into Taxol in a laboratory. The English yew was completely different from the Pacific yew. It grew across Europe. It was common. Its needles regrew every year. You did not need to cut the tree down. This was the key. Meanwhile, in Florida, chemistry professor Robert Holton at Florida State University was working on his own synthesis route. He took the foundation of Potier's work and built on it, eventually developing a four-step process that converted the English yew compound into Taxol with extremely high efficiency. Florida State University signed a licensing deal with Bristol Myers Squibb, one of the largest pharmaceutical companies in the world. Holton patented an improved process in 1992 with an 80% yield, and in early 1993, Bristol Myers Squibb made a historic announcement. They would cease all harvesting of Pacific yew bark by the end of 1995. The forests were safe. In December 1992, exactly 30 years after Arthur Barclay filled that unremarkable bag of bark on a hillside in Washington state, the FDA approved Taxol for the treatment of ovarian cancer. 30 years from a trash tree in the woods to a life-saving drug in a hospital IV bag. By 1994, it was also approved for breast cancer. Today, Paclitaxel, the generic name for Taxol, sits on the World Health Organization's list of essential medicines. It is used to treat breast cancer, ovarian cancer, lung cancer, pancreatic cancer, and AIDS-related Kaposi sarcoma. It is one of the most prescribed chemotherapy drugs on the entire planet. And the Pacific yew tree stands untouched in forests across the American Northwest, still growing, still alive. Still producing a molecule it developed millions of years ago, never knowing what it gave us. That was one tree. Now I want to tell you about a second tree, one that traditional Chinese healers had been using for centuries, and scientists almost missed it entirely. Let us travel now. Far from the forests of the Pacific Northwest, across the ocean to southern China and Tibet, there is a tree that grows here called Camptotheca acuminata. In Chinese, it is called zai shu. Translated directly, that means the happy tree. For hundreds of years, maybe longer, traditional Chinese healers used parts of this tree in their medicine: the bark, the leaves, the fruit. They treated liver disease with it, stomach ailments, psoriasis, skin conditions, and most remarkably, they used it for what they described as abnormal growths, tumors, cancer-like conditions. They did not understand the chemistry, they did not know why it worked, they just knew from generations of observation that it helped. In 1958, a researcher at the U.S. National Cancer Institute named Jonathan Hartwell began cataloging plant-based folk remedies from around the world that were reported to have anti-cancer effects. Zaishu was on the list, it was flagged, but nothing was done with it immediately. Then in 1966, Monroe Wall, the same scientist who had discovered Taxol, was running another large-scale plant screening program. Out of 1,000 different plant extracts tested for anti-tumor activity, only one showed strong enough results to pursue. That one extract came from Paclitaxel acuminata, the happy tree. Wall and his colleague Mansukh Wani isolated the active compound and published their findings in the Journal of the American Chemical Society. They named it camptothecin. Camptothecin killed cancer cells in a completely different way from Taxol. While Taxol works by freezing the structural machinery of cell division, camptothecin goes directly after the DNA replication process. It targets and disables an enzyme called topoisomerase I, an enzyme that cancer cells absolutely need to copy their DNA and multiply. No enzyme, no replication, no multiplication. The cancer cell hits a wall and dies. Traditional Chinese healers had been using this mechanism for centuries without ever knowing it existed at the molecular level. The compound worked in the lab. But like Taxol, it had serious problems in the human body. It was unstable. It was not water-soluble, making it very difficult to deliver safely to patients. Early clinical trials in the 1970s showed serious side effects that halted the research, but scientists did not abandon it. Over the following decades, chemists created modified versions of camptothecin, synthetic derivatives designed to keep the cancer-killing mechanism while solving the stability and toxicity problems. Two of those derivatives received FDA approval. Topotkin, approved to treat ovarian cancer and small cell lung cancer. Irinotecan, approved to treat metastatic colorectal cancer, one of the leading causes of cancer death in the United States. Both are used today, in hospitals right now. Administered to real patients who are alive because of a tree that Chinese healers called the happy tree hundreds of years ago. So what does all of this actually mean? This is the part I really want you to hear. Here is what this story is really about. It is not about secrets. It is not about suppressed cures. It is not about powerful people hiding medicine from you. It is about something far more humbling and far more hopeful than that. Nature figured things out long before we did. The Pacific Yew developed Taxol over millions of years of evolution, not to cure cancer, but to protect itself from fungi, insects, and bacteria. It was a survival weapon, a tree's immune system. And when humans finally stopped burning it as trash and started studying it, they found it could do something extraordinary inside the human body. The same goes for the happy tree of China. Camptothecin was not put there for us. The tree uses it for its own survival. Human beings discovered by paying close attention that nature had already solved a problem we were still struggling with. The National Cancer Institute ran a plant screening program from 1960 to 1981, 21 years. They collected and tested 114,000 plant specimens, over 100,000 samples. From that enormous effort, only a small number produced compounds that eventually became real drugs used in real patients. That is the reality of drug discovery. It is not dramatic. It is not fast. It is slow, meticulous, underfunded, often thankless work, and most of it leads nowhere. But sometimes, a botanist on a routine hike throws some bark into a bag. And 30 years later, a patient with late-stage cancer walks out of a hospital. Today, scientists estimate that 25% of modern medicines are derived from or inspired by plant compounds. Not because of conspiracy, because nature is an incomprehensibly advanced chemist, and we are only beginning to learn how to read what it has written. There are entire ecosystems on this planet, rainforests, deep ocean environments, ancient mountain forests that we have barely studied. Somewhere in those places right now, there are trees and plants and organisms producing compounds we have never seen, compounds that might hold answers to diseases we have not yet beaten. Every acre of forest we lose is potentially a cure we will never find. The next time you walk past a tree, any tree, I want you to think about this. Think about Arthur Barclay hiking through a forest on an ordinary Tuesday morning, throwing bark into a bag without a second thought. Think about Dr. Monroe Wall sitting in a fluorescent-lit lab, going through his 10,000th plant extract, refusing to stop. Think about every scientist who spent decades working on a drug that almost never made it, fighting against environmental crises, supply problems, skeptical colleagues, and the simple, brutal difficulty of turning a molecule found in tree bark into something safe enough to put into a human vein. Think about the cancer patients who enrolled in those early clinical trials when they had no other options. Who said yes to an experimental drug from a trash tree because they had nothing left to lose? They are the reason Taxol made it. The Pacific Yew was called useless. The happy tree was called folk medicine. Both of them were right all along. Science did not suppress this. Science found it slowly, painfully, at enormous cost, and then gave it to the world. That is the story they did not teach you in school, not because it was hidden, because it is too slow, too complicated, and too human for a simple headline. But it is one of the greatest stories ever told.